science for biomechanics as well, I came up with this topic that is if basically science is not open, then it's just a story that you get to tell, nobody get or few get to experience. So it's just basically like a story of you doing something, a research, um, and people are there to either believe it or just not believe it. So that's why it's just the topic. The science is open, otherwise it's a story. Science is always a story, but whose story? I think that's the point that should be here. Okay. So what we're going to talk loosely is about fair data science and what is fair. We'll talk about it, and then a standard that I'm working on actively, which is bits. Then I'll go over sensor placement, which is probably the most relevant to this crowd, and the head descriptions or hierarchical event descriptors. And then I'll end with one example. But before, okay, a little bit about me. I started with biomechanics. I started learning biomechanics with Malihe. I very soon learned not to compete with her at all. She was our top student. I was merely third. And yeah, so I just moved to motor control. And and then after that, in basically like at the end of my undergrad in my master's, and then I moved to neuroscience a little bit in PhD, and then also rehabilitation in the end of the PhD and the postdoc. With that sense, I started with Tehran Polytechnic, which is Amir Kabir, and then went to University of Central Florida, as Manny has said, and then to NYU, which is actually New York Poly, and then UCSD. And I hope that this one is also ending in a polytechnic. So the whole hierarchy would be nice. Um, the research question that I'm now excited about, or I have been excited about since my PhD, was basically how the brain and body responds to the outside factors. Probably because we need to make this a very smaller scope, the outside factors becomes like small delta functions or perturbations. And because I learned biomechanics, it becomes a biomechanical perturbation. So because walking is was hard, or actually like not that exactly, but because I was the first PhD student, I got to work on a seated locomotor task. The second PhD student got the walking part. Um, and this part basically is a recumbent stepper. Assume that is like your left and right hands and legs are basically tied contralaterally, and then you push with your legs and hands. And then there is a perturbation with the servomotor here, while you're supposed to keep your stepping at a constant cue that you see like on the screen. Um, there were EMG sensors as well. We're not talking about them right now, but anyways, so the perturbations are very small, like about 200 milliseconds, and they happen at the mid extension or at the beginning of the extension of each leg. Previously, it was shown that hands basically control the movement. 80% of the energy for driving this device is from the feet. So basically, at the extension onset of the feet of each leg, left or right, um, or at the mid extension of each leg, you get a perturbation, small perturbation. So the interesting part, there are like behavioral parts, but the interesting part was basically replicating a response that we would get during walking and perturbed walking, for example, on a treadmill, which is having a anterior cingulate response. Anterior cingulate is known for monitoring errors during walking. So basically, like here, sorry, I forgot to bring the color bar, but this is a spectrotemporal map. It is called event-related spectral perturbation. So basically, like what when you have, let me change this to a laser pointer. So basically, the x-axis is always a measure of time, Y-axis is a measure of frequency, so it's basically like an STFT, for example, map. And then what you get, if it is red or like a warm color, means that there is a synchronization of activities at this frequency at this time. And if it is blue, you get a desynchronization or less activity than a baseline of this area, for example, at this frequency. So what was previously shown, and I will talk about it a little bit later, and this is just like precursor to whatever I'm going to talk about later on, but is that it was shown that anterior cingulate cortex monitors walking and perturbations and like balance perturbations. So here, balance is not an issue, but still we see the anterior cingulate response. Similarly, or even precursor to that, at like 7 hertz, 3 to 7 hertz, which is called theta band. Similarly, in the supplementary motor area, which receives the anterior cingulate response and then prepares the motor corrections to the motor area, we would see a stronger response or a stronger correction in this area and in theta band as well. The interesting part probably came along, and this is like a paper in 2021 at TNS, sorry. The interesting part was comparing left and right side perturbations. So when it is right side, you can see that probably like this response, and these are anterior cingulates exactly at this band. So basically, if you average this area, this band, and make it a waveform, you get this. That the responses on the right side is a little more pronounced than the left side. So the color bars on down on the bottom shows when there is a significant response, and if it is, for example, the blue one, let's look at the blue one only. Means that it is significantly different from zero for the perturbations, and here you can see that after the perturbation, you get a very significant like anterior cingulate activity similar to what you are seeing here. On the right side, but not on the left side is very similar, but probably like a little wider, so it's like more around zero.
Yeah, yeah.
Um,
So
yes,
what's it happening before the perturbation? Or do you have?
nothing. That's
Okay.
that's the interesting part. So people are just doing assuming the cycling, they are doing their stepping regularly. What at each step we are perturbing. The interesting part is probably this one. So you can see that if it is mid extension,
Mhm.
you can see this. So basically, there is no prior activity, much prior activity. But when the perturbation is happening at extension onset,
Mhm.
people are expecting it, especially if it is on the right side. So basically, you get
So the
yeah.
perturbation was given randomly, or I mean, it's the
No,
you
it's
have a
it is it,
technique,
wa-
or just it was always given at the right,
yeah,
I
it
mean,
was always given
exactly the
at
same time or the angle.
yeah at the same angle. So
So there was
basically,
some kind of like an anticipatory preparation.
only for the right side and only for
That's
one
true.
of the tasks.
Why
Yes.
on the right side?
Yeah. So, I mean, again, that was like for the science purposes, it is very interesting. We had also catch steps. So, yeah, so these are basically like 10 minutes long tasks. One for left side mid extension perturbations, one for left side extension onset perturbations, one for right side extension onset, one for right side mid extension. So four tasks, 40 minutes. The whole process of setting up this person would take about 4 hours, 3 hours, like with all sensor placement and everything. But the whole task was about 40 to 45 minutes. Um, um,
So
another interesting,
fatigue was
yeah,
not an issue.
I'm
Fatigue,
sorry.
fatigue was not an issue.
No,
Same
no.
time.
So No, it's because like the first an hour, first an hour, hour and a half was just prep. And the prep we try to give, it's just basically people are seated and they become mentally a little fatigued, like all tasks with EEG and high density, like the EEGs with large dimensions of sensors, large count of sensors, is always long. But they are not physically fatigued. They are bored, basically, yes. Uh, Fortunately, like the boredom usually doesn't have a response here at these motor areas, or at least like it's not reported. Um,
Sorry about a lot of questions, but the
no, 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 no. That's why I'm
whenever
here.
we
So
deal with the EEGs, and I worry about the level of noise and the artifacts from
absolutely,
movements and
yes, sir.
everything. So especially it looks like you are using a localization tool, which is very
Yes,
sensitive to the noise. So how consistent was it? I mean, between subjects, I
very
guess.
much, very consistent. So, for example, I have 20 subjects, 17 subjects. These localizations are basically we've seen the same components in more than 70% of them. So, so like about 14 or 15 of them were had these components. The interesting part, when you do localization, there are different ways to do localization. So get to the exact, not exact, like estimation of where the activities are coming from. If you do it with ICA,
Okay.
um, ideally, so this was because we were the precursor. I have a whole another set of like presentation slides for how I did it. But if you do it with ICA, you basically get a free noise attenuation or noise separation because ICA is a blind source separation, and it is shown to pick up the biological signals very well. So
Right.
basically, what you do is basically a natural separation happens from the sources that are noise and they are like biological. So that's the part that is very beneficial. But yes, there are lots of ways and tricks that you can do for noise cancellation. I actually did not do much noise cancellation at all, except for ICA and just simple filtering because we had a very good cap, a very good like sensor device. We also had like we just confined the cables very well. At the same time, we had a lot of electrodes, so it's 128. So we had enough dimensions, enough data to separate the noise very well. So basically, like what you see is very much shared across subjects and shared across, yeah. And as long as, and interestingly, like in ERP studies, if you're familiar, usually like the recommendation is to get at least 100 trials and add them together, and then you get just a waveform of voltage. And ERSP, usually like it is the recommendation is very similar, but when I did a significance like study, like how many trials is needed to get a significant response here, it was only 10. So after 10 trials, I would get significance, like the same effect that I'm seeing here. These are all after significance masks, so it is significantly above zero. But my results are very, very robust in that sense. So that was, yeah, but I understand it is a very challenging thing to do. Actually, like my PI and like her PI, which is Dan Ferris, they created a dual layer EEG. So one layer records EEG like from scalp, the other layer records noise from environment, and then they try to subtract it from each other with some algorithms. Okay, so I think yeah, if you liked that topic, probably you want to invite me in another session so I describe and I talk about this. But anyways, let's move forward. But the idea, yeah, I think you get the idea. So you do something on with a mechanical perturbation, and then you want to see what happens with the brain. You can see like the benefits of it, probably like for rehabilitation, for stroke patients, or if for aging, for example, because you see a totally different dynamics for older adults. That was very interesting. Still not published, but it's in my thesis, in my dissertation. hopefully will be published this year. Um, but the problem is that when you have this result, then you want to compare it with walking because my device was a rehabilitation device from design. So, for example, like this friend of mine from Dan Ferris's lab did very similar thing on a balance beam walking with perturbations of a pull from the hip, like pull strings from the hip. And he also saw this anterior cingulate basically response in theta band and supplementary motor area response in theta band as well. Um, so the problem is when it is basically like when we describe these, it is very hand wavy in a sense that we describe it. Oh, like this guy saw this, and this guy, and I am seeing this, so they are very similar. What we are not describing is what are the factors in our experiments that was changed, how much they are changed, how much it was controlled, and can we regress out probably the control factors to see like a more clear response results. I think that becomes very evident when you have two walking conditions, probably one of them balanced beam, one of them like single, like the split belt, and or so on and so forth. If it is like different amplitude of perturbations, or if it is just the same perturbations in two different labs, that becomes, I think that become a challenge, like at least like a question for me at the end of the PhD. And then I just got to work on it at UCSC. So I think that's a motivation, hopefully, for some of you to also at least be interested in in the question of how much my results are interpretable and reproducible if it just goes to whole other task, the same category, and whole other lab. Okay, so with that, basically, like let's go to the main topic, which is FAIR, or just how you can make data and metadata reproducible. This is basically like a paper coming out of 2016 showing that, oh, we need really some guidelines to have a data that is accessible, findable, interoperable, and reusable. And well, you can see that it is cited very well as well. So the whole idea is that any data that you have should be findable, basically like probably having a DOI, and and the authors should be also findable. It should be accessible. So basically, like you should put it on a database or an access network. Basically, like there should be a clear way to access it. It should be interoperable in a way that not just on your PC or on your lab PC. It should be really working anywhere, and it should be very described in a way that it could be reusable. Um, I want to say like reusable slash reproducible, although it is not there. But that's also, I think, one of our main concerns here, how much we can recreate that data again. This is like coming together and with an initiative that like it was there in for EEG. It is called EEG Mini Labs, doing the same task for EEG across like 30 or 50 labs and comparing the results. We are doing very similar things and we call it like EEG many steps or many moves, basically doing similar movement, walking on a treadmill, walking on overground, doing EEG, and then comparing the results. That's like very starting like it started last month basically, and then we are moving forward. If you want to join, you are very welcome to join. So with this, I want to go back just a little. It just seems obvious, probably, or clear that we need the standards and for data and for metadata and how to describe data. And one of the standards that came right after this in probably 2017 is the BITS, or Brain Imaging Data Structure. Basically, it was from Ross Poldrack group. Ross is in Stanford psychology, mostly like data scientists doing data science work on fMRI, and their MRI data is very much like this. Sessions are just names of days and subjects, and It is not at all clear and transparent and fair. So the idea was how should we structure the data, and how which metadata we should add to make this um, reusable for everyone. Okay, so on the storage side, they started defining names. They call it like entities, like the subject, like the task, um, and then also data types. Like if it is a bold is fMRI response, or if it's a T1 structural, for example. And then they also added in directories and then also inheritance. Inheritance means that if you have a task that is run by everyone and they have the same experience, you can have a task description on the top root directory, and it will apply to all of the leaf nodes or leaf directories, for example. And then they also had worked on the file formats and also on making available some converters convert, for example, this DICOM images to nifty NII, for example. This is like for fMRI. The metadata was the same challenge, basically like making a consensus of what is necessary, what is required, or what is recommended, and what is optional, and should be there for each data and for each data modality. And the thing that probably added later on, just like in 2018, was events from our group. Basically, because MRI is not, it's very loose with, can be very loose with um, events. But EEG requires a very precise events because like the temporal resolution is like an order of difference, at least an order of magnitude difference. So basically, we added how events should be annotated or added to this structure. I'm going to talk about this later. Sorry, we are again in the boring part. Um, I want to say like probably the thing that is most related or most relatable here is motion bits. So after the MRI and fMRI specifications, EEG, MEG, IEG, like intracranial EEG, all of them came into like existence. But last year, motion bits came about as well. Is basically to annotate motion capture, just kinematics, and how to annotate that and what metadata is needed. And so basically here you can see like a sample motion capture data structure, and like the colors are basically like what is inside each of these files. The the marker data is on this, basically like a TSV of columns without any title, I think. Um, the next is basically like any data file or any file basically can have a sidecar, which is called the JSON sidecar, which describes what is inside that file. Here, this is a bits convention. For example, here you can see that there is a description of what is this data, a sampling frequency, a name, task description. These are all reserved names. So they are all shared across all data sets. You can build tools to analyze and import these. Then basically you have events. Events have onsets and have durations and have like trial types, for example, kill strikes, if you annotate those, for example. And then you have channels. So you have like different columns here, are each of them are channels and have values. And you describe them here as like the names and what component they are, what is the type of that, and what is the units, and what is the placement of that. The placement, I think, is very important because it is defined very loosely. It is like right foot or right foot on the thigh or something like that. It's just like you can put anything there. And I think from the ad biomechanics and the part of discussion I had with Malihe, basically this is a part that needs to be improved a lot. Okay, so yeah, I think I described most of it. Just I go through it, little points I saw. So because the maintainers or the main people describe developing this were about more on IMU side, they had a thorough defining of channels and coordinate systems and how different coordinate systems should be like basically converted to each other. Because motion capture is usually accompanied, it is a bits. brain imaging data structure, because motion capture is usually like accompanied as like, I don't know, with EEG or with EMG or with something like that. So they added a latency column as well. So you can do a synchronization based on each data point. Um, Still, the sensor placement is a free text field. That's a problem that we should probably resolve, and we are resolving, which we're trying to resolve. If you want to convert your data, you can reach out to me, or I can give you like the maintainer's email. They have tools to convert data, and they will be very happy to help with that. So very similarly, I started an effort for EMG, and EMG can be a little more has become more complex during probably the last decade. So before it was basically like a bipolar EMG, and you can see that motion capture here. This is like one of the last studies I did, data I collected at NYU. Um, Here, this is basically a Delsis EMG sensor, but then you can see that these are also EMG sensors, but they are high density. These are basically 64 channels recording just from one muscle. And from the 64 channels, we usually try to get the motor unit activity bit. That is basically like a combination of the muscle fiber group plus a nerve that activates that muscle fiber. For larger muscles, you have 30 to 50 motor units. So basically, you have here 64 channels. So ideally, you can get up all 30 to 40 motor units here. It's a very cool tool, very actually cheap as well compared to this. Like very similar or even like less priced and pricey than the Delsis system. But the problem is that now the annotation becomes very hard. So how you can describe the system that can describe this one, the bipolar, and also the high density. So that's like a challenge that I'm currently having every 2 weeks, meeting with people around like about 10 people around like with different research focus, for example. And we started from April 2024, uh, and now we have different stakeholders, both academia and industry. Meta got very interested because they are working on a EMG-driven wristband. And then we have like academics from US, from Europe, etc. We will, because EEG is very similar in a signal sense, we will just basically follow the EEG metadata and data, but we will change the metadata mostly. And probably the most, oh, sorry, this is a typo. And the most important ones is metadata that needs discussion is sensor placement and reference. So basically, like these bipolars are basically self-referenced. These ones are these have a extra reference, for example, on the knee for here. So how to describe monopolar and bipolar EMG sensors are right now contentions in the working group. And yeah, I think with this whole thing that we see, like where the electrodes are, the natural discussion is sensor placement. That's what we started with an effort with the motion bits, like maintainers, to see like how we can annotate the markers and sensors on the body. I'm gonna stop this like after this slide and then go and we can look at the specifications here. I think it's pretty cool because I don't know, I'm biased. Um, but we identify 6 requirements for any placement annotation. The body part name, which was the only one that was described in, for example, the motion bids, is the first one. But at the same time, you need an axis name, you need an axis direction, and then you need an axis limits. Then you need a sensor locations as a ratio of the axis limits. So now for example, you define X, Y, Z axis. You need to define how you define the directions. And then you need to define the limits for each body part of each of these 3. And lastly, you need a sensor placement precision. So like
What is
how
the
you
what is
did.
the what is the axis limit here?
So for example, I'm gonna talk. We're gonna just make an example, but here, for example, like for forearm, for example, the axis limits. So assume that we define x as a direction from proximal to distal.
Mhm.
Then the limits in x is from a bony part, a landmark, for example, like the tibial epicondyle, for example. No, not tibial, sorry, ulnar epicondyle here, to ulnar epicondyle here, for example, something like that. So that becomes the limits on that x-axis. The
Oh,
direction is
it's actually
so
okay. Similar to anatomical, just the length, I
it is
guess.
just yeah, it is just anatomical. Yeah, all of
Yeah.
them is anatomical. But basically, it is when when we want to define this and make this, the whole goal is to make it machine readable. So it is clear enough without ambiguity that a machine can read it and understand it. And then if you have multiple data sets, they can be overlaid to each other. So then these are very obvious, seems very obvious, but then usually like missed. I think the sensor placement is also the precision is very important because then sometimes like we do place markers by hand by eye because we feel it is at 50%. And when you go with sometimes like people are more accurate or want to be more accurate, they use the measuring tape and then place it. And sometimes, hopefully, like in the future, not too far, probably people will use 3D scanners to do placement and then record the placement. We do it for EEG actually. So then the whole all of the six can become a structure like this. This is a head structure. I'm going to talk about the head like later on. But basically having a body part, having X, Y, and Z position, and um, X, Y, and Z is defined in a sidecar, in a JSON file for this body part. So it is in the background, and then there is a precision. So I'm going to stop here sharing. Um, and I'm going to share my it's the link that I put. Let me also share the link in the chat if you want to follow. So yeah, let me make it a little bigger. Okay, not very big. Okay, so share this one. So basically, like here, we discuss what is important and what is important for different modality, different sensors, and then defining what is a space, what is reference frames, etc., etc. This is what we talked right now, and like we define placement precision as three levels or hierarchies, and then we have body parts, body, and this like the same thing that we just discussed. But I think the fun comes with a landmark table that we try to define, uh, like for example, an IMU sensor placement as motion capture sensors, and then we try to define any of these sensor placements with this definition, at least like the locations of them. So you can see like on the head, um, on the head, like real head, not HED. So on the head, there is a description, there is an anatomical description, there is a landmark location, so the bony landmarks, and then we define the axes. So here, basically, like you have the coordinate system uh, at the same time. So you have the axes and the axis limits at the same time here. There is left ear to right ear, for example, for x-axis. For y-axis is nasient to inian, and nasient inian is like an anatomical name. You can look it up. And then the z-axis is usually like right hand, but it has a limit. Then with that, when you have defined your x, y, and z, then you can define any sensor location on that body part. Here, for example, for IMU, it becomes like 50%, 0, and 50%. And Yeah, you can probably look at that. It is very long because it is for each body part and for all the sensors that it is there. Um, yeah, so we hope that basically we could be able to make converters for Scenium for different sensor placement schemes for motion capture, for example, that basically you can just, if you put in the fill that it is on the left side, according to this standard, you can basically replace it with a number. And that this number comes back and goes into this X, Y, and Z position. Then you add the precision that you did it with, and then you can report it. And that would be, I think, a much more clear, much more transparent sensor placement scheme.
Okay, so
Yeah,
I have another question. So, do, are you trying to have everyone use the same file later? Is that the goal?
the same
So what
file.
I'm trying to get to is, okay, ISB published like, okay, these are the coordinate systems you should use, and these are the recommendations by the ISB in terms of the marker placements and the segment the coordinate systems and everything. And then the people do not follow those.
Yeah, I
So,
understand. Yeah. So I don't, so we want to encourage. So right now, you can see like the motion bits is have a placement, for example, fill that you can feel and that you can fill. And at the same time, it is a free text. So
okay,
if
so
you want to
you can actually have define the I mean the marker placement for the head at the different place. You will have an options to choose.
yeah, it's going to be an option. So I don't think, to be honest, like right now, biomechanics is not sharing their data very well. So that's the whole, I think, the whole scheme of let's push or encourage and encourage and encourage. So it becomes as much as others. But at the same time, if it is there, if you are going to 90% and sharing your data, we want to like later on encourage that you share it, and we can see that the biomechanics actually like really needs the sensor placement accurately. So we would encourage to just go the next 10%. We will give all required placement tags, if we call it tags, and we just encourage you to just use them. uh, I don't think that it will be used. I try to be realistic. I don't think that it will be used in the next 5 years, but probably after 5 years, we'll be having that. And from the 5 years, I think I can give you an example. So this is let me. This is our data, like database structure. This is handled by us at SCCN. So this site, you can basically, if you go to the Discover, you will see many data sets, uh, many. So, and you can basically like look at how many participants you want to study. These are all bids, for example. But it took about actually 5 years until we have people to actively commit to pushing their data in this format and then coming into this. But then when you have that, probably, oh, this is like a lot. Uh, Probably then you will be able to do analysis on the whole data set. For example, when you have data that is in the same format, you can say, oh, how much of data is good? So how much of data is retained, for example? How much was noise on this data set? These are all EEG. It is not like motion, but just an example of a good data set. What is the gender? And when you have EEG, you have events, what was the events, like what was the major events, so you can do like, I don't know, a point cloud, word cloud, for example, of what was the main events in this data set. So then later on, I want to just show that what happens when um, you have data that then you can do mass analysis. Then you can do, these are all done by our center. not the dataset authors. So we didn't know the authors. We do not know the authors, but we still are able to analyze it automatically and systematically. And we can share the and process data. So later on, if you want to do machine learning, if you do meta-analysis, mega-analysis, you can use our pre-processed data, for example, and clean data. So this is ICA running on that, and basically like all information for that. I think that's a very good promised land in a way that you have data that is transparent, interoperable, and it can be used later on for much more wider examples. I really hope that that becomes the way for biomechanics as well. Um, Let me go back to my presentation and see. It is quite toward the end, so please bear with me here. Okay, that probably like one of the interesting efforts that I started also is a stimulus. So basically, like this is I don't know how much happens for you, but in EEG and like psycho like visual psychophysics experiments, it happens that you see or you do a certain task across different modalities, across different things. Like you watch a movie, you do some perturbations, and then basically it is shared across all many subjects. So basically, the description should not be at the subject level, it should be at the experiment level. So the whole annotation or whole description of what the intervention was should become by itself a standard or a standardized information. That's the part that we want to do. We just started it. Interestingly, we got basically attention or um, mm, contributions from all major modalities that are in bits. There is interestingly there is no little precedent on making a stimulus or just like the interventions standard. And the good thing is that it will facilitate to annotate very, very complex tasks. And very complex tasks can be very simple, like watching a movie. But for watching a movie, there are many levels of annotation that you can do. This is like an annotation of a movie called The Present. But at the same time, what you can do, you can analyze what subjects are in the foreground and background, what is the brightness of the image, what is like the feeling or valence of the subjects that are being played, what are the auditory cues, and what are the shots, like where the transitions are happening. I think I already called the luminance or brightness of the shots as well. Uh, and then you can, if you add eye tracking to it, which this dataset actually has eye tracking, then there is a whole layer of eye tracking that can be fused into this. So basically here, if you wanted to do this on a subject level to describe everything for each subject, then we will have huge events for each person. But the interesting part is because it is shared 2,000 subjects seeing the same image, the same movie, you can just have a event that says, oh, at this time we started the movie and at this time we stopped the movie. And then you can basically describe what was in the movie one time in a different space, and then you can just plug it in here and do your analysis. I think the very, very large implication here is for VR and the experiences that we are doing with VR. But again, we are very at the start. Also, something that is very interesting is like now people are asking subjects to go into the MRI scanner or EEG scanner, give them 10,000, 100,000 images to look at, and then basically managing the whole thing will become a nightmare. So we are working on that too. Okay, um, probably one before last, or probably last, is our effort on head or hierarchical event descriptors. It is how we want to define an experiment, how we define an experiment in a way that it is machine readable and human understandable until it is not. It becomes very complex. The idea is that if I describe it. if you have hill strikes, it happens that usually like the machine that does the annotations, or if you do the annotation yourself, you assign a code for it. For example, number 14, number 10, or just you do a hill strike at a different, like a capital H, or just with space between. So then if you have multiple people doing the same task or similar task, and they want to compare what is happening at hill strike, then you have a problem because they cannot be pulled together very easily, especially across labs. So what happened was basically like our center, Scott McKeag and Kay Robbins, had this idea about 10 years ago. And then basically like what they came up is a structure of vocabulary that is set, that is hierarchical, and a grammar, which is the specifications how to use these vocabulary. The interesting thing with the schema or the vocabulary is that all of the nodes to have an easier relationship. So basically, you can always ask, is animal agent an agent, for example, or is clearing throat a just communicate gesturally action? And you can always ask this and until you go up to the main basically node. Uh, that inference that you can do, that's very interesting because then if you have something that is a gesture communication, but is not the same thing across two different experiments, then probably you can do comparison between them, between two tasks that have a gestural communication with each other and see like how much they have in common and how much they are differ from each other. That's, I think, one of the very, very cool uh, downstream applications here. For example, if you have a mouse button uh, response, then basically what happens, how we would annotate or explain it in head is that the mouse press or left button press is a agent action. So the subject press it is a subject action. It is a press and it is on the left mouse button with a left index finger, for example. That's like how that will happen. Uh, and you can see that already you have like the press already like is not it here is already like, for example, very down. And then the press means an action that is physical, for example, that is doing by hand. So there are multiple levels that press has. And then you can basically assign all of those tags by just saying that it is a press. And mouse button is very similar. It is a human computer interaction device. So there is another hierarchy and it goes down, down, down. And you are assigning all of those tags. It's just saying that it is a mouse button press. So this is good, but where is the application? And I think I have this probably for the end. So this is the data set that will go on hopefully today or at the worst by the end of next week because by the end of this week because I need to present it on Saturday at SFN. It is 2,600 plus participants, 2 terabytes of EEG data and behavioral responses. It is 6 tasks, 13 total runs. Runs means like a task that has run repeatedly. And it has passive tasks. People are watching movies or resting state, but then are tasks that you respond with a mouse button, as I said, clicks. So this is a massive data set. It is from Childmind Institute in New York, Healthy Brain Network project. So you can see that now it becomes very useful to have those head tags because you have many different tasks. Interestingly, this task has EEG, but also it has MRI. So then similar tasks, for example, like movie watching ones, are done with different modalities. This also has eye tracking as well. We are not releasing eye tracking right now. Probably one more interesting part is that the kids, these are like 5 to 21 years old, this 2,600 subjects, also done a full comprehensive psychometric questionnaires And then they they did a factor analysis on that to make it out of like 100 questions, make four factors: how much attention they had, how much externalizing, internalizing, and P factor. P factor means like a comorbidity of different psychological problems. So, like if it is attention deficit, for example, and then P factor, interestingly, like is not very related. But if the person is hyper hyperactive or um, doing externalizing a lot, then the P factor is very important. So now you have a data set that is very rich. You have also psychometric values. Now, what is ready next step is to do inference between the psychometric factors like this and EEG and the behavioral responses that we recorded. Interestingly, like to make this data available, it was a long journey, about a year and a half that I did. The data was there, it was raw, it is already public, but it was not ready to load easily. And it required inspecting the EEG data, adding the events, adding behavioral data, doing quality checks, and then making sure those are, to just share those are all with the available with all metadata and data that we just discussed. So now that data set is ready, you just need to about 50 lines of code or even less than that to load the data. Actually, loading the data is just one line, but load the data, do a full analysis, ICA, filtering everything, and get your results. For example, the Earth plots that I showed you. It is very short. At the same time, we have that data on Neuroscience Gateway available. It is a free high performance computing resource. So basically, you can just load the data there, do analysis, and get the results back in less than a day on all 2,600 subjects. The good, interesting part is, probably I end with this, is that it is fully annotated with head, because so now you can do all mouse button presses can be regressed out or analyzed across tasks. And the unwanted benefit that we had is that the tasks become very transparent. So when I wanted to create a figure for this tasks for the paper, I actually give the head descriptions of the tasks to an LLM plus the event table and ask it to create this figure. This figure was created by an LLM. Basically, like the part that you see on top was what was shown to the subject. And I asked the LLM with just the head tags to recreate these plots. And it just created it. It was very, very, very nice finding. Um, the this one, like the down panel, is the timeline of when these are presented to the subject, and I just give the head descriptions plus a snippet of the events, and I ask it to create a Python code that plots this part with also like with these tags, and it just created it. It's very similar to this one, also the same, asking it to create what the subject saw and the timeline. And even when it was a multiple like levels of things that are happening at the same time over each other, when I just gave the tasks descriptions, the head descriptions plus a snippet of the events, the LLM could just recreate it. I want to say that this is basically probably the ultimate transparency, in a way that if you describe the tasks, an LLM can understand it and reproduce it and making a figure for it. I think that was the goal. That we didn't have that goal, but now it has become our goal to make experiments like that. And yeah, thanks. That was the end. Yeah, I had a thank you as well. But yeah, sorry, it was boring, but yeah.
Seems like you were
If you,
doing more mechanistic work before, but now dealing with a whole bunch of informatics work. So,
yeah,
so what's your plan?
Okay, yeah. So right now, actually, I'm I'm resuming. So basically, like, it is a US way of if you work on whatever you funding that you have, basically. So, but at the same time, I become very passionate for this. But now I'm moving back. I have moved back to the my research questions right now, and the experiment that we are starting is actually doing a bimanual control. Like the cooperation of different limbs is very interesting, and it is very pertinent to aging actually. When you have like multiple limbs, like the coordination between them becomes very impaired, very much impaired very fast with aging. So we are doing bimanual control experiments for controlling, for example, a weighted bar, and then asking people to manipulate that. We're doing EEG and multi-scale EMG, which is interesting. Is just basically like doing EMG like with small sensors and with high density sensors and the goal overall is to do a brain source level connectivity analysis to EMG source level motor unit activities. Basically seeing how much of the muscle activity is coming from the brain and how much of the muscle activity is not coming from the brain, because we have different sources of um, basically like motor coordination or efferent flow, which are not from the cortex. They can be subcortical. That is, I think,
Okay.
my research field right now. So yeah.
Okay, so spinal level control.
Oh, it can be a spinal. It can. Oh, I want to say
It
it
is
is
very
cortical and
hard
sub.
to. I mean, I'm sorry about all these questions. I'll shut up after this question.
No, 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 no,
But,
no, no.
but
That's a good
it's
question. Yeah,
really really challenging to differentiate the where the neural signal is coming from because when you say like subcortical, what do you mean by subcortical? You
yeah.
know,
That's
this. I mean cortical level control also receives the signals from the spinal cord level, and they are all intermingled around.
yeah,
So it's very hard to differentiate there where the
yeah.
source. So you
So
have EMG signals, but I mean it is in a sense impossible to differentiate where the signals are coming from.
I can differentiate what is cortical. I cannot differentiate what is subcortical or where. So if I know what is cortical, I hopefully know the rest is subcortical. But when where subcortical it is, yes, you're right. That become a challenge.
But
There are
depending
people
on
who
where
try to do
where you
TMS. Yeah.
Yeah, depending on where you actually record the neural signals. I mean, alpha motor neurons receive signals from the spinal level control too. So if you are recording the neural signals at the alpha motor neuron level, then it's hard to tell if it is coming from uh, the upper brain or from the spinal cord.
Why? If if I
For
let
example,
me
just
let me
muscle
push
stretch.
back.
Muscle stretch that actually has an input to the alpha motor neuronal pools as well. But it seems like
Okay.
it's going to be very challenging research.
Yeah,
But I
but
think that's
yeah,
very very necessary research actually.
yeah, I agree. I mean, for my research, at least at least I said it for myself, is that I'm not doing invasive anything. So, but the interesting part is that um, right now the composition to motor unit alpha like motor unit activities, motor neurons, alpha motor neuron activities is kind of possible with 90% accuracy. And then what I can get is a basically like a very robust quote unquote causal relation between the um, motor, like the cortical activity and the motor unit activities. Basically, like about probably 20% can be described in that realm. 80% of motor unit activity cannot be described in that. and that becomes the next step, which right now, actually, I started to learn that people are doing. I don't know if you're familiar with TMS, like the
Hey.
stimulation that you do with the brain, and then the hand moves. But then people are doing it with the spine as well. And